And it's my pleasure to present another oak study. I think there is still something new to learn from this study. The title is Breeding Sessile Oak, Quercus petraea for Drought Stress Tolerance. But I'm going to present the results of a cooperation project covering genetics, genomics, physiology, as well as a common garden. So, the acronym of the cooperation project is AQUAREL. It means adaptation of Quercus on relict sites. And we focus on sites where oaks are autochthonous. We are talking about steep slopes and screes, where we have mostly no beech competing the oaks, so we hypothesize that our oaks are autochthonous there. We hypothesize also that there is a long habitat continuity. And given that we mostly have extreme sites, we also think that there must be a local adaptation to drought stress. As I already mentioned, we have a genomic and a stress physiology approach to address this question. In order to address the question of autochthony and of the relic character, we have, on the one hand, a genetic approach. On the other hand, we have some indicator species of barks and mosses and lichens. As an outcome of the project, we recently established a progeny test which we expect to be able to convert into a seedling seed orchard in future. And yes, I would like to show you the main results and discuss a bit the perspective in terms of breeding. So these are the main questions we have. We first address the question where we have such potential relic stands of oaks. We compared many databases about habitats, Natura 2000 and other protected areas. Then we investigated fauna and flora, mainly subproxilic beetles, which function as indicators of habitat continuity, or mosses and lichens, which often indicate lack of disturbance. Concerning the origin of the oaks and the genetic variation, we address the questions where do the oaks on such sites originate from, and are they autochthonous? And we also investigated inter- and interspecific genetic differentiation, but today I'm going to focus on sessile oak. Concerning the local adaptation, as I said, we had a physiological approach and a genomic approach based on pool sequencing. So this is an overview of our study populations. Our study area is in southern Germany and covers also the easternmost part of France, Alsace. We had a total of 63 study stands. 50 of them were such putatively relict stands, all of them on dry sites. And as a comparison, we used also 13 productive sites on more humid, growing under more humid soil conditions. Each of them closely situated to one of the dry relict sites. And Quercus petraea was the most common species. For our genetic approach, we had 8 chloroplast microsatellites, which were useful to address autochthony and the refugial origin of the populations. We've seen already some of these graphics showing the post-glacial recolonization. It's these markers we used. And we also applied 22 nuclear microsatellite markers, which were useful in order to identify the species, but also to study the intraspecific genetic diversity. Study of indicator species of flora and fauna was restricted to 39 relict stands for the subproxilic beetles and 34 relict and 10 productive stands for mosses and lichens, but I'm gonna skip this. We had, maybe I can mention, we had many rare, very rare species, so-called Urwald-Reliktarten, primeval relict species, which indicate a long habitat continuity in the case of beetles. So, and this brings us already to the first results concerning autochthony and the relict character. Here we could observe the known post-glacial recolonization pathways, but we could also observe a very low intra-population variation. Although I have to say we went beyond the standard and we genotyped 24 individuals per stand. And we have here a comparison with managed stands. I forgot to mention that those putative relic stands are being unproductive. They were practically unmanaged, or maybe function as pastures in the past, or only for fuel wood. And here are the results. We have 0.1 haplotypic diversity within stands. If we limit the comparison within the upper Rhine Valley, where we have a good data set of managed population, we can see the difference. We have 0.055 in the putatively relic stands. We have here 0.125 in old managed stands. Old managed stands are mostly originate from previously copy stands. And we can see the effects of seed admixtures in recently established stands, on those stands that were established after the Second World War, and also under a different management regime, because during the 19th and 20th century, formerly coppice forests were gradually converted into high forest, and there was often plantation at these sites, at these forests. And this is the result of structure with our 22 nuclear microsatellites. This was useful, among others, also in order to form species poor pools for our pool-seek approach. And what we see here is that Quercus petraea, this sky blue, represented with a sky blue color, is the most common species. We had 45 stands, which we classified as Quercus petraea. We had 4 Quercus pubescens stands. We are here at the northern limit of the distribution range of this species. We also had 2 Quercus robust stands and 12 populations which were admixed. On this map, I'm showing the result of an intraspecific structure run within Quercus petraea for k equals 2 for 2 assumed subpopulations. What we see is a slight east-west gradient, but genetic differentiation was very, very low. We had an FST of 0.08008. In order to address local adaptation, we had a genomic approach based on pool sequencing, and we had mostly relic stands, but as I mentioned in the beginning, we paired some of them with some moist, with some humid sites situated closely to the relic stand. I already talked about the first steps up to species identification, and after characterizing each individual taxonomically, we formed equimolar pools and carried out next generation sequencing. Our approach was targeted sequencing. The Quercus robur reference genome was used, and we targeted all protein coding sequences. The total target size was 30 megabase pairs, and we had an average coverage of 200 times and an average read length of 300 base pairs. In order to confirm that our pool-seq approach works well, we carried out a validation experiment where we both individually sequenced two populations, and we also carried out pool-seq and compared the estimate of allele frequency. And as you can see here, this was quite successful. We had a coefficient of determination of 0.93. This compares the frequencies measured by individual sequencing versus those frequencies estimated by pool-seq. And we had also a procedure here, including quality trimming, read mapping, and after various steps, which we can see, but I wouldn't like to get into much detail, we could map 73% of the reads, about 120 million reads, against the reference genome of Quercus robur. And these data were used both for an outlier approach and for a genomic environment association, for a GEA approach. Here we can see the paired population consisting of one dry and one relatively humid site in the vicinity. And this is the genetic differentiation, the heat map here of FST generated with about 1.4 million SNPs using pool fstat. It seems plausible as most pairs cluster together, and we can see again that the genetic differentiation is very low, equaling 0.004. And here we can see the results of the outlier test, of the FST outlier test. We had in total 2,377 outliers, quite many, but anyway, I'm relieved that I saw similar situations in previous presentations. Here is a list with the annotation of the top 10 outliers we had, and we decided to map the allele frequencies, to put them on a map to see how they behave. Here we have two examples. This protein tyrosine kinase, PTK, it plays a role in abiotic stress. It's an important part of the signaling pathway leading to stomatal closure. And we can see an almost consistent pattern of one allele being a bit more frequent on the relict site and less frequent on the relatively humid site, which is, however, inverted in two cases. And this is the case also for the other SNP residing within a genome area encoding geraniol 8 hydroxylase, a secondary metabolite. So I have to speed up. 3, okay, this is good. Thank you. Okay, this is not exactly the same approach. This is a complementary approach, including 45 Quercus petraea pools, and here we associated environmental conditions with genomic variation. Here is a short list of the 14 environmental variables we used. We used topographical variables based on the digital elevation model, longitude, and some climatic variables from Workrim and Chelsea, as well as soil nutrient class. We've seen such graphics too. We can see that there is not always a good correlation of different approaches, but we decided to follow a linear, an LFMM approach. And these are the top 9 significant associations we had. Aspect was the top factor based on 3 approaches. We have here 1,586 SNPs, which were associated with aspect based on the LFMM. And a further remark, 5 out of 9 SNPs were located in chromosome 2. I mentioned already also the physiological approach. For the physiological approach, there was a common garden with progenies. There were 7 stress metabolites which were assessed, and similarly to the outliers, there was not always a consistent pattern where all dry sites behaved the same way compared to the humid ones. A very brief summary of the results is that there are different strategies of humid and moist sites, which were not completely consistent, but in general, oaks on humid sites, their progenies accumulated 2 tannins, vescalagine and castalagine, which were consumed under drought stress, while the progenies of dry oaks, so to say, were able to produce these tannins, these phenolic compounds, also under drought stress. This is a very brief summary of the result. And as I said, as a final result of the project, we collected acorns in autumn 2020, and we established a progeny test with open pollinated families from 12 stands, 5 from each stand, and from the remaining stands we had bulk seeds. First results show that there is some clinal variation and ecotypic variation with mountainous populations, with progenies of mountainous populations as expected flushing later. And the same was the case about the northernmost populations we had. And this brings us to the summary. We saw by means of flora-fauna indicators and genetics that we have highly autochthonous population. We saw signals of adaptation, both using genomics and physiology. These signatures are quite complex, I would say. And the perspective is, and what we aim is to continue with backward and forward selection within these plots we established, which can be converted sometimes into a seedling seed orchard. Thank you for your attention. This was quite a lot, I think, but yeah. Glad to answer questions.
Thank you very much, Charalambos. Do we have any questions about breeding? There.
Thank you. I'm interested in the outliers. You had quite many outliers, and you said some of them were in coding regions. I don't remember the number, but was it rather more or less than you would expect from, or was it under or overrepresented? Did you check this?
I'm not sure I can, I didn't have a certain expectation because of my limited experience on this topic, and I have to say the credit belongs actually to Devrim who did this analysis, but my impression was that we had really many outliers. On the other hand, we had more than 1 million of SNPs, so 2,000 is also, yeah.
Sorry, I mean not the number of outliers, but the number of coding SNP outliers within the number of outliers. Maybe we can talk later.
Maybe you do it like this, yeah.
Thank you.
Any other questions?
Very nice talk. You mentioned the market edited selections. You will do market edited selections. What do you mean?
What I mean is that we have the possibility in future to select individuals within families and within provenances, and also select provenances in this test, in these common gardens.
Yeah, but in forestry, the last 30 years, We have done many marker-edited selections, but never successful. No company and no breeding program took up the marker-edited selection, because usually you get markers from one family, one province. They are not transferable to another family, another provenance. So now everybody move into genome-wide selection, genomic selections. That's whole genome-wide, population-based, not a marker-edited selection. Marker-edited selection are very successful in crops because they have major alleles, major genes, but not in trees. This is just the results last 30 years.
I rather don't expect to exclusively select by using genomic results, but rather phenotypically select, because we have 4 sites. The one is on an extreme dry site. I rather expect that the results of the physiological analysis and genomic analysis will rather assist, but I have something more classic in mind for the future.
Okay.
Okay, any more? I think we say thank you, Charalambos, and move to the next.